Thank you very much, Alessandra, for that very kind introduction. I did change, however, the title a little bit just to focus on some bits of the work that I wanted to share with you today and highlight. I'm hoping that now you can see my screen. So what is really exciting for me today as well is that actually we've put this work as a preprint and it came out just today. So this is actually the sort of like opening of this work to the public. So I'm really excited to get questions and really ask anything because, you know, it's still work in progress. So it's really good to get some feedback on it. And so basically, in a nutshell, this is just a transcriptomic single-cell atlas of the human developing spinal cord. And the idea here is that the spinal cord is one region from the central nervous system that extends from the brainstem to the lumbar region of the vertebral column. And the spinal cord is this long, thin, tubular structure that is made up of nervous tissue. And it allows for movement, feeling, sensations, and control of bodily functions. And together and next to the peripheral or the central nervous system, we can find as well the peripheral nervous system, which is here labeled or depicted in blue. And in this case, the peripheral nervous system is the one that carries the sensory information about the external world and also the body to the brain and the spinal cord. And the morphological arrangement of the peripheral and the central nervous system has been extensively characterized and studied over, you know, the past, I would say maybe the last century. And this is one of the cartoons, a famous cartoon from Santiago Ramón y Cajal, where one can see basically a cross-section of the central nervous system here in the middle, but also a small overview of what the peripheral nervous system looks like in a cross-section. And this morphological arrangement is important because it is the way that the neural circuits are assembled. And this molecular and physiological neuronal diversity that emerges within this structure is generated in a characteristic spatial and temporal manner during development. So here, what I'm showing you here is some developing spinal cord cross sections of human embryos that we characterize when I started my postdoc. So what you can see here is the same structure that will later on be formed, but just at early stages in human development. So very early on, if you look at the neural tube, the first thing that we see is these cells that are labeled with SOX2, which are progenitors, and these sort of flanking regions highlighted here in pink, which express islet 1 and HV9, which are motor neurons that over the course of differentiation of development, you can see it here with these different numbers, which correspond to the Carnegie stages of human development. So these are stages of embryo development in humans, basically differentiating and populating these structures here, which are known as the ventral horns. So over time, what we observe is this transition from progenitors to neurons. And specifically here, what I told you is that we see the differentiations and the generation of motor neurons, which are those that innervate the muscles. So most of the information that is available for, in terms of spinal cord development is coming from studies in model organisms. And as of today, limited analysis had been performed on the relevant stages of human development. And for example, in my case, some of the characterizations that I did was just by looking at my favorite genes in specific sections at specific time points in human development. And one of the reasons why doing this in human is slightly more complicated than doing it in model organisms is the lack of access to the tissues. So with the aim to comprehensively characterize the diversity and complexity of cells within the tissue, we decided to perform single-cell mRNA sequencing in the human developing spinal cord. So what I'm highlighting here are the 4 stages that we were able to collect. So these correspond to CS12, CS14, 17, and 19 that correspond or take place over the week 4 and up to the week 7 of human development. So the samples, they were obtained from elected abortions from the HDBR resource. This is the UK Human Developmental Biology resource, which provides the samples for then you to do your investigations. And what we did was that we collected the samples, we dissected them, dissociated them, and sequenced them at the Crick using the 10X platform. So this is a single cell sequencing technique that is droplet based. And the idea here was to profile the gene expression in cervical and thoracic regions at the 4 different stages to follow this spatiotemporal differentiation dynamics. And we concentrated in these stages because they cover the major events in spinal cord development, namely, so progenital patterning, dorsoventral patterning, neurogenesis and gliogenesis. So initially from these 4 samples, we recovered around 70,000 cells in total. And in a first step to visualize the datasets independently, we perform uniform manifold approximation and projection, UMAPs, for dimensionality reduction. So what you can see here is that this generated basically 4 different UMAPs, each corresponding to one of the stages that we analyzed. What's also evident here is that we were able to clearly identify clusters that were evident in every single of the samples. So some of them are color-coded here, so you can see how we got progenitors and neurons from the central nervous system, progenitors and neurons from the peripheral nervous system, but also some other what we would call unwanted cells, such as mesoderm, skin, and blood, or others. And by others, what we mean is cells that have very poorly resolved transcriptomes, and therefore we couldn't really associate to any specific tissue that we know of in the embryo. And from this analysis, there were very obvious differences in the proportions of cell types from different time points. So for example, most neurons were contained in the datasets that were obtained at later developmental stages, which is consistent with the idea that initially in developing, or in any embryo, you first have progenitors and subsequently you have neurons or differentiated cells. In contrast, mesoderm cells, so labeled here in brown, were enriched more in early stages versus the late stages. So in this case, mesoderm cells are in close proximity to the nervous system cells. And in fact, they are the tissue that originates, for example, the vertebrae. So for instance, dissecting this cleanly as we were collecting the samples was more complicated in smaller embryos than in bigger embryos. But nevertheless, since what we wanted to do is generate a spatiotemporal gene expression atlas of the developing neural cells, what we did is that we selected the cell clusters in each stage and generated a combined dataset. So this is what you can see here, how the combined dataset looks like, and this is basically our kind of working dataset. So the combined UMAP contained in total around 43,000 cells, and it allows us to clearly separate both progenitors and neurons from both tissues, so the central nervous system, but also the peripheral nervous systems. What was good to see as well is that cells from replicate samples, whether they were from the same or different sex, they intermingled well within the embedding, suggesting minimal or at least a good correction of batch variation across embryos. And something that I think is already evident in this UMAP is that in terms of gene expression similarity, neurons emerging from both cell types projected to adjacent clusters, which I think points to a convergence in gene expression programs of central and peripheral nervous system cells. Next, what we wanted to do is to classify all this diversity of cells that we generated. And for that, what we did is that we used a trick that we've used before for a mouse single-cell RNA-seq transcriptomic atlas that we published a couple of years ago. And the trick here is that we use a binary knowledge matrix. So the idea here is that with the information that we have available in the literature, we build a matrix in which each progenitor, domain, and also neuronal class is identified by the expression of a characteristic marker set. So once we did this, what we can do is that we can select the cells that we've assigned to different identities, and we can plot the resulting average levels of marker expression of these cells that we assign the specific identities. And this is what you can see here in the case of progenitors. So now what we have is human cell types that we've assigned to, and that express the genes that we know are characteristic for these specific domains and types of cells within the developing spinal cord in this case. Something that we're also very keen to do was to compare the transcriptional similarity between progenitors and neurons of mouse and human. And for that, what we did is that we used expression levels of homologous genes in each neural cell type in mouse and human. And what we did is that we estimated the Pearson correlation coefficient for each pair of cell types. So the first thing that was really good to see and to validate is that in fact, mouse and human cells are assigned to the same identities correlate very highly to one another. So this is sort of represented here with the diagonal. And also this also allowed us to see that in terms of progenitors, their similarity seems to be higher in progenitors that are sitting adjacent to one another, which again is consistent with the knowledge that we have on the gene expression program of these developing progenitors. By contrast, what was also kind of, I guess, unexpected to see is that if we looked at the neuronal cell types, they weren't really clustering according to their spatial location, but rather into their functionality. So basically, excitatory and inhibitory neurons were clustering better together in terms of gene expression similarity than just looking at the spatial patterns. Something else that we could do is that now we could look at our genes of interest and ask how many of these genes are showing novel expression patterns in human compared to mouse. So one of these examples, which is something that we were anticipating to see, happened with PAX7. So PAX7 is a gene that normally would be used to assign cells into these DP domains, which is basically dorsal progenitor domains. And this was very useful for both mouse and human species. But now, so human here is the first column and mouse is the second column within every gene. But now, if you look below here, in this region, which is called FP, that stands for floor plate, what we detected here was the expression, what it seemed like increased expression level for PAX7 that was never ever seen in the mouse data set. And this is something that we saw in our data set, but also was already pointed to us by Kate Story's lab a couple of years ago. And now they've actually published some work on it, in which when they stained for PAX7 in developing human embryos, they already identified PAX7 being expressed as you can see here, in the human floor plate. And as I was saying, this is a specific expression from the human that we couldn't really find, or we cannot really find in the mouse. So with the data that we had, what we could do now is select the cells that we assigned as human floor plate and ask which are the genes expressed in the floor plate cells that highly correlate with PAX7. So we did this, a number of genes pop up. And what was nice to see is that some of these are not really expressed in the mouse, suggesting that perhaps the expression of this gene, at least is bringing up some novelty into the human developing spinal cord. Also, I think we found that it was worth mentioning that these genes that are expressed in the human floor plate and that they seem to follow the pattern of expression of PAX7, they're also identified in other domains. So by no means we're saying that these genes are novel targets of PAX7, but perhaps they've gained new functions in this new territory, so the floor plate. So another difference that we identify from the very few differences that we identify between mouse and human corresponds to the gene NKX6.2. So in this case, this is a gene that we normally use to characterize and profile progenitors that correspond to the P1 identity. So these are progenitors that will give rise to a specific type of interneuron, the V1 interneurons. And what I hope that you can see here is that when we looked at their expression in the human data set, we observed that the levels of expression for the human, for this gene NKX6.2, were highly significant compared to the mouse, which, in which we failed to detect this expression. So we wanted to follow this up further. So actually, it's kind of interesting in the sense that for NKX6.2 in the mouse, the first time point that really we assayed in the single cell mouse transcriptome atlas was 9.5s. And as you can see, NKX6.2 expression, so in red here, in E9.5 embryos is contained and is expressed only in the P1 domain, which is what I was telling you before. What is kind of nice is that earlier on, this gene seems to be expressed more ventrally. And this is highlighted here by this expression, basically the yellow expression of the gene, which means that it's co-expressed with NKX6.1 and is expressed at more ventral positions. So because we saw this sort of dynamic expression of NKX6.2 in mouse from ventral to dorsal, even though it's not dorsal, it's more intermediate positions, we wanted to see if in our human time course, we would also see dynamic expression of NKX6.2 in ventral domains. So to do that, we just got the cells that we assigned, or the cells that we assigned to different categories, to different progenitor types, and looked at the expression of the gene at various time points. So what we observe is that yes, NKX6.2, it is indeed expressed in what we assign to as P1 cells, but when we look at other ventral domains, namely the PMN domain and the P3 domain, what we observed is that NKX6.2 was also expressed in these cells earlier on. And what was even more striking is that at late stages in the human spinal cord development, we were also even able to detect this expression in P3 cells. So now just to put you in context, the 9.5 mouse stage would correspond to the CS12 stage. So this expression at these really later stages in development suggests that there's been a shift in the temporal dynamics of this gene in human compared to mouse. Now we wanted to validate this, and for that, what I'm showing you here is just an immuno where I'm again showing you NKX 6.2 restricted expression in the mouse in the P1 domain. And now when we look at the equivalent human embryo, and the way we did this is by performing RNA scope. So this is just by looking at the transcripts of the NKX 6.2 gene. Yes, we detected it was expressed in the P1 domain, but also I hope that you can appreciate how the expression is also spread all throughout the ventral domains that we can find in the developing human spinal cord. So as I was telling you, there's definitely a difference in expression. And because this type of pattern, we observe it at earlier stages in the mouse, we call this to be a heterochrony. But also what I told you is, what I told you as well is that at late stages in development, we observed that the NKX 6.2 expression was retained in these particular ventral cells, which are called the P3 cells. And what was really good to see and exciting is that when we performed the RNA scope in this human CS15 embryo, so that would be kind of like in between when it is expressed at high levels and at low levels, what we observe is that now NKX 6.2 is expressed still in the P3 domain, and this is something that we never see in the mouse. And one of the other differences, this is the difference that has been previously characterized, and that it is believed to be human specific, is the fact that the motor neuron domain in humans is quite broad compared to the mouse, and we'll talk about this slightly later. But also, there's this region in the PMN domain, so in the blue domain, that contains or that expresses at the same time the NKX 2.2 gene and the OLIC2 gene. So these are the cells that are basically labeled in pink. And what has been claimed to be specific is the fact that there's co-expression of these two genes in this big population here of cells. Because when you look in the mouse, it's actually very rare that you detect these two genes being expressed in the same cell. And I'm pointing you at the probably like only cell that you can see that has that gene expression pattern within the mouse developing spinal cord. So now this is kind of interesting because the fact that you express OLIC2 and NKX 2.2 in the same cell may mean that these cells are sort of changing their identity. So on the one hand, these cells that express the two genes, this is why they're pink, they could be cells that are sort of transitioning, so they're becoming more ventral progenitors, so becoming P3 cells, and that's changing into red, and therefore becoming a different type of neuron. So P3s generate V3 neurons, but PMN cells normally generate motor neurons. But the alternative hypothesis that also this sort of co-expression raises is that the specific expression and combined expression of these two factors could give rise to a sort of different cell type, which is called oligodendrocyte precursor cells that gives rise to oligodendrocytes. So basically what this would be telling us is that these cells over here, instead of giving rise to neurons, they give rise to glia, which is the cell type that is sort of alongside with the neurons protecting them. And this hypothesis comes from the fact that in the developing chicken and mouse spinal cord, when we look at when are these two genes being co-expressed in the developing spinal cord, we find that this happens at late stages in development. You can see it here in yellow. So the idea is that the moment that these two genes are getting or are becoming expressed at once in the developing spinal cord, there's a switch from progenitors generating neurons to progenitors generating glia. And I tried to sort of like explain it here with a cartoon for you. So the idea would be that initially you have cells in the green domain that express OLIC2, and these progenitors over time generate neurons, motor neurons. But then over time, these cells sort of start to express OLIC2 and NKX 2.2 together. And then they sort of switch fate, and instead of being neural progenitors, they become oligodendrocyte precursors or progenitors, and thus they give rise to oligodendrocytes. So we were really interested in knowing if this co-expression in the human that came up really early on was perhaps meaning that the cells that we identify in the human are in fact maybe oligodendrocyte precursors rather than motor neuron progenitors. So the only thing really that we could do with our data is to look at the gene expression of these cells in comparisons with cells that only express one of the genes and try to see if that was representative of them having more of a gliogenic function. So for comparison, what we used is the cells in the mouse that appear at late stages in development, but that also co-express the two genes. So what you can see, and I think that, so my favorite example here is FAP7 because it's the one that is highly expressed. So here in the mouse, it's only cells at the latest time points that they express FAP7, therefore they have the oligodendrocyte potential coming up, and that is what is characteristic for the mouse. However, in the human, when we look at FAP7 expression, it seems that these cells already at the first time point that we use for the human data set, they already express FAP7. Perhaps suggesting that these double positive cells have more gliogenic potential at early stages compared to mouse. And this is something that we also try to measure here by using a gliogenic score, which basically the idea here is that you select for genes that you know that have a function in your tissue or your cell type of interest, in our case, the gliogenic score, and then we compare it with the levels of expression in one of the given populations. And what we saw is that for the double positive cells, this score is higher. So perhaps suggesting that these cells have more gliogenic potential. And the other thing that we were really excited about doing is thinking about the rate of neurogenesis. So I told you that previously we had done some immunos in which we were able to sort of characterize mouse and human development side by side. It helped us to sort of match the time points accurately and also sort of look at the dynamics. And something that we observed but we really didn't quantify is the fact that the motor neuron domain in human is way broader than the comparable domain in the mouse. So now, because we had characterized human development in many cell types within the central nervous system, but also because we had the temporal resolution, we could now ask how are the dorsoventral progenitor domains evolving over time in the human, and how does this compare to the mouse? So to do that and to simplify the data, what we did is that we grouped all the dorsal domains into what we call the PD domain, so the dorsal domain, progenitor dorsal domain, then all the intermediate fates into PI, and then we look specifically into the motor neurons and the P3 progenitors in both mouse and human. So here human is, so mouse is the dashed line and human is that corresponds to the solid line. So if we go back to the motor neuron domain, that is the one that was expanded in human already at, and I forgot to say that we did match the mouse and human samples 1-to-1 according to our previous characterization. So if we look at the motor neuron domains in human, we can already observe that very early on, as I was telling you, the domain is expanded. And we identified that this was also the case for the P3 domain. And these domains sort of shrink without time, which is something that we were, we will also be noticed by basically doing immunos. What instead we hadn't observed before is that now this increase in the size of these domains is at the expense of the intermediate and the dorsal progenitors, suggesting that the rate of neurogenesis is sort of overall similar between mouse species, between mouse and human species. However, there's a higher initial proportion of ventral progenitors in the human developing spinal cord. Something else that perhaps is worth mentioning and studying further is that when we look at the expression of PD progenitors over time, we didn't really were able to capture sort of like the peak of expression of the dorsal domain, at least that's what we think. So perhaps what this suggests is that the dorsal progenitors persist for a longer period of time in human compared to mouse. Now, the other thing that this single-cell RNA-seq allows us to do is to reconstruct gene expression dynamics along the differentiation trajectories. And in order to do that, we performed RNA velocity analysis. So the idea here is that RNA velocity enables the recovery of directed dynamic information by using splicing information. So what it does is that it estimates the reaction rates of RNA transcription, splicing, and degradation, and it infers if a gene is being induced or repressed. So that's giving you an idea of directionality in gene expression. So what we're showing here is the trajectory inference analysis for the progenitor motor neurons towards motor neurons. And what we see is kind of very simple. So progenitors are differentiated into neurons, and this seems that they follow sort of a single trajectory path. And what you can see is here, if we plot it by time point, the earlier time points, they seem to have more progenitors, and then the latest time points seem to have more neurons, which is really expected. But also what we could do is estimate a latent time. So the idea here is that you can reconstruct the sequence of transcriptomic events, but what you can use is ground this information on the transcriptional dynamics and take into account when a gene is normally overall expressed within the differentiation trajectory. So I'm showing here the smooth expression of this trajectory inference for the neurogenic trajectory in human and mouse. And what you can see is that when we look at how this transition happens, we see that progenitor genes are going down over time. Then sort of neurogenic genes start to pop up. So this is, for example, the case of NeuroD4. And then later in development, you do get the expression of neuronal genes, such as in this case, MNX1, for example. And we did observe this, that both, or the same thing for both mouse and human. So basically what we could say is that the gene expression dynamics for mouse and human by trajectory inference is remarkably similar between the two. We also reconstructed the trajectory inference for all of those progenitors and neuron domains that had, or in which we had enough cells to perform the analysis. And here I'm just showing you an example of how this looks like if we compare it between human and mouse. So in these two first rows, what I'm showing you is a progenitor marker, SOX2, and a neuronal marker, level 2. And what you can observe is that SOX2 is expressed earlier in the latent time pseudo time course, and a level 3 is expressed at later time points. And for human and mouse, it's more or less the same in each of one of the domains. And now if we, for example, concentrate here on genes that are specific for each of these sort of domains in the progenitor area, we can see that they are normally expressed at early stages of the differentiation trajectory, and that the dynamics, they are not that different between human and mouse. And finally, because now we have information about the in vivo human relative development, we could make a comparisons with the differentiation of motor neurons in vitro from human embryonic stem cells. So we've previously developed a protocol that has allowed us to really follow and recapitulate the dynamics or temporal expression of this differentiation over time. And what I'm just showing you here, and I just want to highlight, is that we have an RNA-seq that we could use to compare with the single-cell RNA-seq. But in order to do that, what we did is that we selected highly expressed genes that varied during neurogenic differentiation from the daily bulk RNA-seq experiments in vitro. And what we then did for the comparison is generate a pseudo bulk from the in vivo human data sets. And we did this by averaging the gene expression of progenitors and neurons in all ventral domains from the data sets in every single time point. Then what we did was a pairwise comparison of the in vitro and the in vivo time points. And what we observed is that the CS12, so this is our earliest time point in vivo, corresponded to our in vitro differentiation from day 4 to day 6. Then our latest time points in the in vitro differentiation corresponded to latest embryos, so for instance, for day CS14. And then for the latest time points, even though there's a somewhat higher correlation that at earlier time points for the late time points in vitro, we didn't observe this like really remarkable comparison or correspondence between one time point or the other. So what we can do now is think about how long does it take in vivo and how long does it take in vitro. So if we think of, for instance, of the stage CS12, so that corresponds to the gestational days between 26 to 30 in human development, and in our in vitro, as I told you, is from day 4 to day 6. Now, if we concentrate on the CS14 stage, so this would be the second stage, this corresponds to days 31 to 35 of human development, and in our in vitro to day 9. So what I find, well, and then here it's a bit more complicated, but definitely these stages, they correspond to higher numbers than 9, even though we cannot really assign any, I would say, comparable equivalents. But what I think it's really nice here is that we can already observe that between our in vitro differentiations from day 4 to day 9, there's a 5 days time difference, and which is kind of surprisingly similar to the actual in vivo correspondence. So now this is obviously a very gross comparison, and perhaps we should be more careful in how we phrase this, but I definitely say that our in vitro temporal profile and is perhaps not that far away from what we see in vivo. So just to finish, I just wanted to tell you that today what I tried to discuss with you is the fact that there's an overall similarity in the composition and dynamics of the developing neural tube, that we weren't able to identify that many differences, even though we detected some differences between mouse and human. And specifically, we perhaps because we were looking for, but we definitely found some heterochronies in the case of NKX 6.2. And also, I think that we were able to show that in vitro motor neuron differentiation from human stem cells is able to recapitulate the differentiation tempo, so the speed of development from in vivo. And I just wanted to finish by showing you this small movie that we made, because I'm really happy that we've put all this data together and available for people to do some research on it. So here you can find the web app. And basically the idea is that people use it as a resource in case they're interested to see, if the gene of interest is expressed at a particular place in the human data set, essentially. And we have 4 time points, we have tons of cells, and we really hope that you use it. And as I was saying, the preprint just came out today. So please just, go to bioRxiv and look it up. And I'll just finish by acknowledging everyone that I've worked with, essentially. So James, I'm a postdoc in James's lab, and Rory, who's a PhD student who's helped me with the analysis, a lot with the analysis, and Chris Barrington from BAS, who has been instrumental for the developing of this project. The advanced sequencing facility, I would also like to acknowledge them, their effort, because normally these samples would arrive to us no earlier than 5 in the sort of evening, and they would stay with me and help me out with the 10x equipment and pipeline. Mary helped me out with the RNA scope technique, and Mark Pollitt was actually the one that made the web page available to everyone. Then obviously the previous work from the mouse single cell RNA-seq was also very important for this, to be able to do this work in human. The whole Briscoe lab, because they're amazing in terms of feedback, and Kate's story, because since the very beginning she was sharing all her knowledge in the human field and human development field with us, the funding bodies. And yeah, and thank you for listening. And I guess that I'll take some questions later.